Great. So here we have some test data, and let me see. So the test data just basically, oh, that's not what I want to do. Let me actually make this bigger. So the test data consists of two genomes and a reference database. So for a good reference database, I would recommend RefSeq, but Binder has limits, and we have a time limit here, so I don't want to spend too much time. So what I did is I just found a bunch of mycobacterium genomes, and I concatenated them together, and I made a database out of them, a diamond blast database. So Pseudo-Finder can run diamond blast and regular and conventional blast, depending on how fast you want this done. And I'm going to use diamond here because it's much faster. So Mycobacterium leprae is what causes leprosy, and it's an intracellular pathogen. And it has a very interesting genome, is that its genome highly resembles the genomes of recently acquired endosymbionts, in that nearly half of its, a big part of its, actually I don't know exactly what proportion of its genome is pseudogenes, but it's a lot higher than average bacteria. And Mycobacterium tuberculosis is it's another mycobacterium. It causes tuberculosis, and its genome is not in the same state as the genome of leprosy. I'm not 100% sure why Mycobacterium tuberculosis avoided such a huge, the genomic dilapidation that's occurring in Mycobacterium leprae. My going hypothesis was that it's because it's not an intracellular pathogen, but I'm actually not sure right now whether tuberculosis is an intracellular or extracellular pathogen. But for the purposes of this demonstration, we're going to assume that this is intracellular and this is a good reference genome. This is actually the most closely related genome to leprae, and we're going to use that as a reference to look at the selection. Okay, so let's print the Pseudo-Finder help menu, and we're going to see that there's a number of commands that we can run. There's the annotate, re-annotate, visualize, DNDS, and test. So I'm not going to go over visualize and test. And test is something that you can run on your own system when you download the GitHub repository for Pseudo-Finder, because this requires some files that are not present in this Binder tutorial. The re-annotate module is pretty nice because if you run annotate, and it takes a while if you run it with like RefSeq, for example, and you want to alter some parameters, but you don't want to have it redo the entire blast, you could just run re-annotate and give it the output files generated from annotate, and it's going to rerun with different parameters, and it's going to be a lot faster because it's just going to use the previously generated blast results. So Pseudo-Finder help, we want to run Pseudo-Finder annotate. This is the main command of the script, and let's see what we have. Okay, so we have some required arguments. This is the genome that you want to annotate. This is the blast database, the reference database, and then the out prefix. Threads, this is how many threads you want to give this program. So the more, the faster. We only have one allocated here on this binder, so we're not going to touch this. Intergenic length, this is something that I mentioned in my in the slideshow, where if you wanted to not look at intergenic regions that are very short, like 30 base pairs, you could set this to be 100 or 1,000. Length pseudo, this is also something that I mentioned. So if you wanted to, so the default here is 65%. So if a gene is less than 65% the length of a reference homolog, then it's going to be labeled a pseudogene. So we could set this to lower. or higher if you want to be more conservative. Shared hits, percentage of blast hits that must be shared in order to join two nearby regions. So this is related to the fact that I should actually explain this one first. Hit cap. So when Pseudo-Finder runs blast of your query genome against a reference database, it's going to generate a number of hits, number of homologs. So you can alter the number of homologs that you want to use as a reference to evaluate whether a gene is a pseudogene. So the default is 15, but you could set this to be higher or lower. And to go back to shared hits, this refers to the percentage of blast hits that must be shared in order to join two nearby regions. So this is, I feel this is pretty self-explanatory. So if you have 15 blast hits and 10 of those blast hits, so if you have two gene fragments that have been separated by a stop codon, and each gene fragment recruits 15 blast hits, and all of those blast hits are exactly the same between the two genes, then Pseudo-Finder is going to assume that they are the same gene that have been separated by a stop codon. But if only 2 of the 15 blast hits from each gene are shared, then it's not going to assume that. If less than 50% are shared, it's not going to assume that. If more than 50% are shared, it's going to combine them as potential fragments separated by a stop codon. So this is something that you can alter as well. And you can say that you can be more conservative and say that if 80% of the blast hits between two adjacent fragments need to be shared in order for Pseudo-Finder to call it a pseudogene. E-value, this is for blast. So E-value is how stringent you want to be with the blast search. Default is 10 to the minus 4. You could be more stringent than this, and you can say 10 to the minus 10, just to recruit something that's more closely related. Distance, so if you have two adjacent regions that share blast hits, but they're separated by some space that a gene finding program might assume is intergenic, that might cause problems with joining those adjacent fragments if the intergenic region is too long. So that length, you can set that the distance here. So if the two gene fragments are separated by more than 1,000 base pairs, then Pseudo-Finder is not going to join them, even if they share 100% of the blast hits. But you can set that lower, and you could set the threshold to be 2,000 or 3,000, or you could set it to be 100, however low you want, depending on how stringent you want the joining of the potential gene fragments to be. Contig ends, right? So if you have intergenic regions at contig ends, those could also be included in the analysis. By default, Pseudo-Finder does not look at those regions. And then intergenic threshold, number of blast hits, right? So I talked about this during the slideshow. So if you have some intergenic space that's blasted against the reference data set, how many blast hits does it need in order for it to be annotated as a pseudogene? Right, so it's calculated as a percentage of maximum hits allowed. So if maximum hits is 15 and default is 0.3, that means you need at least five hits to call a particular intergenic region a pseudogene. Right, so these 6 flags are in reference to the PAML loop, or the selection part of the analysis. So if you provide a reference genome for DNDS analysis, then it's going to use that genome as a reference for DNDS analysis. These flags, I mentioned these during my slideshow. The -DNDS flag will set a lower threshold for assigning pseudogenes based on DNDS value. You could also set the max or minimum DS, right? So if two genes are really distantly related, you might have a really high DS. So this is going to limit how much genes can actually be, how many of the genes in your data set can actually be used for, can actually be compared for. So basically, DS is the rate of synonymous mutations that have occurred, and this is essentially a measure of divergence time. So if you have two genes that are highly divergent, then any calculated DNDS value might not be useful. So by default, this is set to 3, and the minimum is set to 0.001. So you do want to stay within that range of DS values to have a reliable DNDS estimate. Because if you have a really, really low DS value, this basically means that genes are really similar, nearly identical. In that case, the DNDS value might not be as informative. But if you have a really high DS, then that might also be result in a really non-informative DNDS value, because the genes are just too divergent, where you might see a lot of mutations that have occurred that are synonymous, and it's actually you've saturated all the potential synonymous sites within the gene where mutations can occur. Right, diamond, right? So if you want to use diamond instead of blast to do your analysis, this is going to have the program run a lot faster. And dash skip is actually this is something that's deprecated, so I'm not going to talk about that. Okay, so let's run a command. So just copy and paste from the walkthrough on my GitHub. So we're going to give it the Mycobacterium leprae as the genome of interest. The database is going to be the Mycobacteria, and we're going to set the out prefix to test annotate. Enter. Okay, so it's running blast. Oh, whoops! Actually, I just realized that I forgot to add the -- diamond flag. So what we're gonna do is we're gonna remove annotate. Then we're gonna rerun with -- so that it runs a lot faster. All right, any questions so far while this is running?
Okay, so I'm removing this test annotate, which was a directory that was written by the last command.
Yeah,
Okay.
I don't think it matters. Like, I think Pseudo-Finder will overwrite the directory. It doesn't. I know some programs like Unicycler or they don't, or Proca, they don't like overwriting, so they're gonna crash. But in this case, it won't do that. Okay, so we see that it finished already. So let's look at the output. So it generated a bunch of files with this test annotate prefix. So it created these two files, the proteome and CDS. So these are essentially, the proteome is just the amino acids, the gene call, the amino acid sequences from the GenBank file that was provided, and CDS are the coding regions in nucleotide. format. So if you look at this, you see the genes, nucleotides, and the proteome here, amino acid format. And the same thing for the reference data set. Let's see, what is this here? Oh, I guess okay, that's not here for some reason. It also outputs the intergenic regions in nucleotide format. We have those there. We have the BLAST output, if you want to evaluate that yourself. And then we have the pseudogenes outputted in FASTA format, the nucleotide sequences. And then we have GFF files. And these GFF files are a really important outputs because they will actually tell you which of the genes are pseudogenes and why. So let me look at this file again like this. So here we see, well, let's look at the log. So the log actually summarizes the output. And we can see that all our parameters are summarized here. We have the input, number of ORFs. So this is not the entire Mycobacterium genome. This is just a subset that I extracted for the purposes of this tutorial, just to have it run faster. And this is the number of ORFs, initial ORFs that were analyzed. And then total number of pseudogenes from these ORFs is predicted to be 247. And we have a breakdown of why what the pseudogenes are. Essentially, we have 37 genes that are too short. We have 46 genes that have been fragmented, and 164 have been predicted to be within intergenic regions. So this is something where whatever gene predicting software was used to generate the GenBank file, let's say Prodigal, wasn't able to detect an open reading frame of a particular gene, where when you blast it against the reference data set, it recruited a certain number of hits, and that told Pseudo-Finder that it's a potential pseudogene. Right, so when you look at the pseudos file, the GFF file, we see that all the pseudogenes are summarized here. So this one, for example, it has the general breakdown of a GFF file, but the last column essentially gives you all the information you need to know about why that gene is a pseudogene. So here the reason is given, that's predicted fragmentation of a single gene. And then here as the last field in this, you can see that the GenBank locus tags that went into calling this a fragmented gene, and you see that there's two intergenic regions that were actually recruited the same blast hits, and they're adjacent to each other. So this told Pseudo-Finder that this is likely it used to be an ancestral gene that got broken up and is not even recognized as an open reading frame. Another example here, this one, pseudogene reason the open reading frame is about 60% of the average length of the hits to this gene. Right? So it's a lot shorter. So there must have been a deletion or something else that happened that broke this gene up. And then the functional genes, or the intact genes, we're planning on renaming this functional to intact, because we really can't assume function based on this, but we can just say that the gene appears to be intact. And this looks like a GFF file that you might see from Procon. Okay, so as you can see here in this log, we didn't include a reference genome, so it didn't run the DNDS analysis. So here we have 0, but what we're going to do now is we're going to rerun the program, but we're going to give it a reference data set, a reference genome. We're going to give it this Mycobacterium tuberculosis genome, also in GenBank format. And the out prefix, we're going to call this test annotate dnds. We're going to run that. So this might take a little bit longer to run because it is going to have to align the files. It's going to have to make code on alignment. It's going to have to run code ML. But that shouldn't take too long. So the output from this is going to be really similar to the output we've seen from the previous run, except we're going to see an additional directory that's outputted, and we're going to see a little bit more pseudogenes predicted based on the fact that some of the genes in Mycobacterium leprae appear to be highly divergent relative to Mycobacterium tuberculosis.
Yes, as soon as we're done, I'm going to look into the pathophysiology of leprosy versus tuberculosis. Yeah.
Yeah, that's really interesting to me.
Yeah.
Because
I guess we should be calling it Hansen's disease now, right? Rather than leprosy.
Why?
That's I guess that's a more that's what the medical community calls it now.
Oh, is it?
So I guess that there's historical negative connotations with leprosy now or something like that. But
oh yeah.
Hansen disease or leprosy, I mean.
Yeah.
But yeah, I have no idea why one would have more pseudogenes than the other. There's definitely different evolutionary trajectories. But then again, I'm like you. I don't know that. I don't know anything at all about their pathophysiology, so I should have a look.
It's really interesting, and one thing that you want to consider if you are running Pseudo-Finder and you provide a reference genome, you have to really think about that reference genome that you're providing. Is it really a good reference, right? So the reason I'm providing Mycobacterium tuberculosis here is because it doesn't appear to have a lot of pseudogenes. If you run Pseudo-Finder and you just run it with refseq, it doesn't appear to have a lot of pseudogenes, and it's relatively closely related to leprae. But it does have some pseudogenes. So when you're thinking about comparing genes from leprae to tuberculosis, that's something that you have to consider. Is it a good comparison? Are there going to be pseudogenes in leprae that might also be pseudogenized in tuberculosis? Because if that's the case, then you might see a pseudogene. You might have a pseudogene in leprae that actually, does not appear to be highly divergent from an ortholog in tuberculosis, or it might be really highly divergent, but maybe not because it's a pseudogene, because it's adapting to something. So that's why when we're looking at the endosymbionts genomes, we really want a good reference that we know is free living, because we know that free living bacteria don't have a lot of pseudogenes. They can't afford to keep a lot of pseudogenes. So when you're thinking about what's a good reference data set or a reference genome for leprae, you really want a free living bacterium, because free living bacteria can't afford to have a lot of pseudogenes. They have really low pseudogene densities, and this Mycobacterium fall into that category. I don't really remember exactly how many pseudogenes it has, but that's something to consider because it is a pathogen. So it still might be undergoing some kind of ecological shift that's relatively recent. So the genome might not have time to have sorted itself out yet, and it still might encode some proportion of genes that are pseudogenes. Okay. So this just finished, and we see here a quick summary of what was the selection, the DNDS analysis, and we see that it found about 1,400 genes within the DS range, the recommended DS range of 0.0. Actually, it should be 0.0013. Change this. Average DS among orthologs within the specified range is 0.8. and the average DNDS is 0.15. Okay. So when we ran this program, we went with the default DNDS cutoff of 0.3. So anything above 0.3 is going to be predicted to be a pseudogene. So let's look at the output file. So as you can see, there's there's more pseudogenes that were predicted here relative to the last run, and same numbers for short, fragmented, and intergenic pseudogenes. But we also have an additional 76 genes that are added to this because they have elevated DNDS values. So if you want to look at evaluate the DNDS output a little bit more, you can go into this output directory that's created. And then the summary file is here. All the blast results, the summary file is here. And this basically tells you the ORF from your query genome, the reference match in the tuberculosis and the reference genome, the DN value, DS value, and then DNDS value, and then N or Y, whether it's a pseudogene or not. Right, so if you go into this DNDS analysis script, you'll see that you can actually look at the codon alignments yourself. You have the query gene sequence, right? So this query gene sequence is going to have the query gene and its homolog, right? So you'll see this aligned as well. Want to look at the alignment, you can inspect the alignment. You can inspect the codon alignment here. And then you can also look at the raw output from CodeML here. Whoops. Okay. It's going to give you codon usage. It's going to summarize all that for you. It's a pretty nice summary. And it's going to give you a bunch of values. Of these, the important ones are DNDS, DS, and DN and DS. Okay. Right. All right. So all of this is just the subset of genes that are shared between Mycobacterium leprae and Mycobacterium tuberculosis. So depending on how close related your reference genome is to your query genome, you're going to have more or less genes here. That's why for the core annotate module, you want to provide something like RefSeq or NR. That's more of a comprehensive database, so you get a higher coverage of the genome that's annotated. Okay, whoops. So I'm gonna wrap it up, and if are there any other questions?
Can you give it more than one genome as a reference?
No, you can
just one.
You can concatenate files into a single FASTA file or a single GenBank file.
Yeah,
there's a way to do that, but I think that generally it's accepted that if you're doing DNDS analysis, you want a one genome to one genome comparison, because if you have another genome that you're providing within that same file, you're going to have slightly different relatedness to that genome. So divergence time between the genomes is going to be a little bit different, and that's going to that might have some influence on the final DNDS calculations, because DS is going to be overall DS is going to be different.
So with like everything else, this sounds like a trial and error, right? Because perhaps there's a lot of close relatives, but choosing one versus the other, like you said, is going to imply differences in divergence time, right? That will affect your results. But I don't know, like choosing a bunch of them can basically show, right? If like how much of a factor that plays on your results. Okay.
You can rerun the DNDS module by itself. So if you Um, so for the DNDS module, you can just provide it with, actually, let me try running it now. But you can rerun the DNDS module. You don't give it GenBank files, you give it protein sequences and then amino acid sequences, amino acid sequences and nucleotide sequences of your query, and then amino acid and nucleotide sequences of the reference. And it's gonna just basically do an analysis and provide you with just this one directory that will have the, just this output. And you can run this on multiple genomes and then compare the output and see if there's any patterns, right? So if you have one query genome and you have 10 references and you have one gene that's constantly always looks, that it's under neutral or positive selection, high DNDS relative to all of them, then that will add to your confidence that something's going on with that gene.